Good afternoon, everyone. Welcome to the third webinar in the Young Scientists Program webinar series. Just for as an update for the ones that are arriving for the first time, this is a series of 10 webinars that will take place between May and September this year in preparation for the annual IUPAB Congress that will be hosted along with the Brazilian Society of Biophysics and the Brazilian Society of Biochemistry and Molecular Biology that will take place in October this year because of the pandemic in a virtual format. So this kickoff event will bring together leading scientists in various research fields of biochemistry and biophysics, and they will be seconded by young scientists that were selected from applicants from all over the world. So these meetings are taking place every Wednesday at 2 p.m. Brasilia time, and today we'll have a talk in the theme of plant biotechnology, biofuels, and bioenergy. So before getting to our program, I would just like to make a advertising that there are fellowships for students and young postdocs to participate in the IUPAB meeting. So the interested people should go to the web page that is shown here in the screen, and then you can find information how to apply for these fellowships. And there is a fair amount of money that IUPAB has provided to be distributed as fellowships for people to participate in the meeting. So if you apply and you are selected, you'll be exempt for paying the registration fee. So I really invite you all to apply for these fellowships. So moving into our program, so we have today three speakers. We have Dr. Igor Polikarpov from the Institute of Physics of São Carlos at University of São Paulo. He's going to make the main talk, and two young scientists, Adebare Adeleke from Nigeria, from Modibo Adabama University of Technology, and Robson Tramontina from Unicamp, Brazil. So I would just like to make a introduction of our main speaker. So Dr. Igor Polikarpov, he has a degree in physics from University of Belarus, also a PhD in physical and mathematical sciences from the same university. And after completing postdoc training at the Max Planck in Gesellschaft and the Edinburgh University, he moved to Brazil, where he stayed for a few years at the National Laboratory of Synchrotron Light, where he collaborated in the construction of protein crystallography beam lines. He's now then he moved to the Institute of Physics of University of São Paulo in São Carlos, where he's currently a full professor. Dr. Polikarpov has his main interests are in structural biology and molecular biophysics and their applications to bioenergy and biotechnology. He has almost 300 publications in peer-reviewed international journals, which are highly cited. And he's very active in the field of promoting bioenergy research, being involved in the coordination of several collaborative projects with European Union and other countries in South America, and also in India. And he's presently the coordinator of Polo Terra, USP São Carlos, which is a thematic pole devoted to renewable energies and environment. He's also involved in mentoring a lot of students, being involved in the creation of the inter-unit PhD program in bioenergy that congregates 3 universities in São Paulo. And he has a large experience of supervising PhDs, masters, and also postdocs. So without further ado, I invite Dr. Polikarpov to start his presentation. It's going to be a 40 minutes presentation. And I'd like to, people interested in making questions, you can either write your questions in the question and answer tab, or you can raise your hands, and then after the talk, we're going to open your mix, and then you can make your questions orally. So Igor, please, I'm going to stop this slide, and then you can put on your presentation.
Thank you very much, Eduardo. Let me try to share my screen again. Okay. And it was like that.
Perfect.
Can you see my presentation?
Yeah, it's full screen.
Cool. Okay. Well, before anything else, I would like to thank the organizers of these seminars for inviting me to give and to present you some results of our recent research in the field of enzymes related to active, again, conflict carbohydrates. Field of research is stemming from the idea that our energies should gradually become more and more renewable. And one of the ways to produce renewable energies and green chemical compounds is to take plant biomass, treat it, and enzymatically hydrolyze. And then by fermentation or catalytical upgrading, one can produce fuels or green chemicals. And that will be a renewable process. We can pull more biomass and transform more fuels and green chemicals. It was particularly successful in Brazil. We have at least one example of commercially successful plant, Raízen built a second generation ethanol facility in Piracicaba and increased ethanol production to millions of liters per year, starting from plant biomass. And it was so successful that the company now openly says that they're planning to build more units, which will replicate the technology and produce more and more second generation ethanol, which would be really good. Well, there were a number of initiatives in the United States, Europe, and elsewhere focused on second generation ethanol. Why Raízen was so successful? Well, the answer is here. Basically, one of my friends used to say, I don't know what the question is, but the answer is money. And that's exactly what you can see from this slide. Raízen was the most cost-efficient project, and which is great. I mean, we have a number of very efficient technologies in Brazil, but for us to be a leader in the world leader in one of the fields is really something exciting and not always frequent. What is the process? Well, the process is based on sugarcane first generation ethanol production which was established in Brazil in the 70s. And you basically collect sugarcane, you squeeze it, and you end up with sucrose, which you can ferment into ethanol or just transform into sugar. And also piles of fibrous material known as bagasse. Well, those of you who have sharp eyes probably can see this man standing in front of the truck. Can you see the guy? So it means that that's a full-scale heavy-duty vehicle and not a toy truck. And that's a really huge pile of bagasse. Since bagasse has been transported into industrial location already, you already paid for the transportation costs. So that's a great advantage of the technology in Brazilian settings. And if you look at the plant biomass, such as sugarcane bagasse, it basically contains three parts. Cellulose, which is quite simple polysaccharide made out of glucose. Hemicellulose, which is more diversified class of polysaccharides, which contain a lot of pentose sugars, and lignin, which is biomolecule, macromolecule, full of aromatic rings. And at least the first two components can transform into monomeric sugars and then ferment catalytically upgrade for further use. And by doing so, you add value to these compounds. It looks very simple. In fact, it's not, because in addition to optimizing your treatment process, you have to develop and apply optimized mixtures of enzymes which will hydrolyze these polysaccharides. And even if we look at the cellulose, which is simple, although quite recalcitrant polysaccharide, we need a set of enzymes to degrade it into monomeric sugars, which will include endoglucanases that will act predominantly on amorphous parts of the cellulose, biohydrolases, which will processively transform polysaccharide chains, cellulose chains, into cellobios, and beta-glucosidases, which will transform oligosaccharide into glucose. Within the last 10 years, it became apparent that oxidative process realized by lytic polysaccharide monooxygenase is also very important in this whole process, and this additional layer became more and more studied and used in the enzymatic transformation of the plant biomass. If we look at this process from perspective of enzymes, it actually shows, in addition to what I said already, applied importance, something else. Actually, due to the next generation sequencing technologies, we have an exponential growth of the number of sequences of the enzymes in general available for biotechnological exploration, studies, biochemical studies, biophysical studies. Now, while a number of sequences was growing exponentially, our capacity to solve structures and to biochemically characterize enzymes basically was increasing linearly. And that means that there is a huge and growing gap between number of sequences available, a number of enzymes which have been biochemically characterized, and number of structures solved. In 2013, year for which I have statistical data, we had about 5.5% of the enzymes biochemically biochemically characterized and about 0.5% characterized structurally. From that period of time till today, the situation only became worse because the number of sequences continued increasing and the number of structures was not growing with the same pace. It means that we have a huge opportunity to find new genes, to find new enzymes, and to study them, and eventually to use them for possible biological and biomechanical applications. Within the field of cellulosic ethanol and beyond. And I will give some examples of what we were doing and what I think we can do in the future. So we'll start with a few short stories. Obviously, when we realized that there was a tremendous opportunity in solving structures, we went and solved some structures, trying to understand why enzymes work the way they do. And we started, one of the first structures was endoglucanase from the GH family 7, glycoside hydrolase family 7, from fungus known as Trichoderma herzianum. It's actually a close relative of Trichoderma rhizii, which is most widely industrially used for biomass degradation. And to our surprise, this endoglucanase had actually much higher activity in beta-glucoxyloglucan, lichinon, and xylon than on crystalline or amorphous cellulose. That was sort of unexpected, and we didn't know how to interpret it first. So we decided to solve this structure and look at the structure. So we crystallize the catalytic domain of the endoglucanase and look at it and compare it to the structurally homologous enzymes. And by looking at the active side and solving the structure with the investigating the structure with the ligands and substrates, we were able to explain why this enzyme is so promiscuous and has activity at different polysaccharides. We also looked at the cellulose from the same family. Actually, for those who are not familiar with the enzymes, GH7 family is the main family for enzymatic degradation, fungal enzymatic degradation of cellulose. This family contains cellulose and endoglucanases. Depending on fungal species, it can be one enzyme of each class. It can be more than one enzyme. For example, Mycelioptera thermophila has 2 cellulose and 2 endoglucanases. One of them has a cellulose binding module appended to the catalytic domain. And I don't have time to explain why it's important, but it's important. And the reason why this fungus has 2 enzymes of each family, one with CBM and another without CBM, probably reflects in its robustness toward enzymatic degradation of plant biomass. So the difference, if we look at the structures of these 2 enzymes, they're very similar actually. The difference is in loops, which cover the active side. In cell biohydrolases, the loops are long and form sort of a tunnel in which the cellulose chain is shredded and catalytically cleaved. Now, endoglucanase has much more open structure and will recognize cellulose chain, but will not processively move on the surface of cellulose, not most of the time at least. When we cloned and expressed and purified enzyme, we looked at its characteristics and because Mesothelioptera thermophila is known to be a thermostable enzyme source. And indeed, if you compare a thermostability of Mesothelioptera thermophila to a much more studied Trichoderma rhizae, you see that the former enzyme is more thermostable. And if you measure residual activity, you will see that Mesothelioptera thermophila is actually quite stable at 60 degrees, at least for 10 hours, whereas Trichoderma rhizae dies in less than an hour or so at 60 degrees. So, and running temperature, running catalytic reactions on high temperatures is actually an advantage because you have less contamination, you have faster catalytic rates normally, and you have improvements in quality, so that's good. Then we use small angle X-ray scattering technique to have a look at the overall structure of the enzyme and also have an idea about its mobility. Also run molecular dynamic simulations to try to explain the low resolution envelope of the enzymes. It looks like a tadpole, actually, the enzyme. There is a big, huge, quite big catalytic domain and a small cellulose binding module joined by a linker peptide. And in most of the fungal cell biohydrolase, this linker peptide is glycosylated or glycosylated, and that adds to flexibility of the enzyme. Actually, this enzyme has two main subpopulations in solution, and because it has two hinges in the linker region. And then we produce catalytic domain and crystallize catalytic domain. We were not able to crystallize the whole length enzyme. Actually, nobody solved as yet the whole length enzyme because linker peptide is very flexible. So we crystallized the catalytic domain, and then we solved it with the uptake and products, and that allowed us to understand how enzyme liquefies cellulose and also run molecular dynamic simulations, trying to gain an idea about molecular flexibility of enzyme. Actually, it's quite a bit different in flexibility compared to Trichoderma rhizae, because Trichoderma rhizae stays closed all the time and has a tunnel-shaped enzyme, and that's why it's very expensive. Now, Mesothelioptera thermophila opens up quite a bit because of the specific mutation in this loop position. And that makes the loop more flexible and also explains a smaller processivity of the cell biohydrolase, which is good in some applications. And we also run statistical coupling analysis of this protein, and that allowed us to select specific polyvolve sectors within the protein structure and compare these sectors to the steps in catalytic mechanism of the enzyme. And actually, they quite strongly correlate to crystallizing catalytic activation steps of the cellulose action. So we said, okay, well, we have some idea about endoglucanases and cell biohydrolases. What about lytic polysaccharide monooxygenases? And we set out to characterize the two-domain LPMO9H from Thermocellomyces thermophilus. It's not a new fungus, it's the same as Mesothelioptera thermophila, just microbiologists changed the names of the microorganisms already that we sometimes have difficulties to accompany these states. Lytic polysaccharide monooxygenases are monocopper enzymes, and they help cellulases in degradation of cellulose. There are a number of them nowadays in CASI family, in CASI database, belonging to the families A, 9, 10, 11, 13, 14, 15, 16, so far. All of them have copper coordinated by histidines. One of the histidines is N-terminal histidine. That's why it's somewhat more difficult to produce the enzyme. They're very stable. Actually, you can heat them up and then you cool them down. They re-nature, and that's good. That's not very common. So what we started by doing by when we decided to explore this enzyme, we looked at the activity of the enzyme, right? And we realized that it has activity against PNPG and PNPC, which are synthetic substrates for glycoside hydrolases. And unfortunately, it did have activity against PNPG, which indicated that our preparation was just contaminated with acidic glucosidase. Quite common when you express enzymes in fungal species, simply because fungi, they produce cellulases normally. And what we did, we made use of the stability of the enzyme toward temperature. So we basically heated it up to 85 degrees, hoping that all possible GHs and other enzymes will be dead at this temperature, and then cool it down and measure the activity. So basically, when we did it, the additional activity on PNP, P was gone, and we were very exploring the enzyme. So one of the things which we did, we looked at the specificity for the substance, and it's known that if LPMO is activated in the absence of enzyme, it will produce hydrogen peroxide, and in so-called futile cycle. And so we said, okay, well, if it will produce hydrogen peroxide in futile cycle, if we add the substrate, then amount of hydrogen peroxide which will produce probably will be smaller. And that's exactly what happened. If you add phosphoric acid swollen cellulose, which is an amorphous cellulose substrate, the hydrogen peroxide production dropped quite significantly. The same happened with xyloglucan, but other substrates were more or less ineffective in hydrogen peroxide suppression. We looked at the pH dependence of the enzymes, actually quite a bit more active at pH and basic pHs and alkaline pHs, but the problem is that it becomes so active that it will oxidate itself and dies in a very short period of time. We also looked at the possible activators. We have to reduce LPMO, and it can be done, for example, with ascorbic acid in yellow. And when we do so, we see oxidations at C1 or also C4 positions by the LPMO. Well, we need to supply co-substrate, which is oxygen or hydrogen peroxide. We need also to reduce enzymes. So following a couple of previous works, we said, well, maybe we can use light for doing so. So we took chlorophyll and ascorbic acid and shine light at the chlorophyll. If you do this, you will see that LPMO is quite strongly activated. In the dark, it does not do much, but in the presence of light, it does. It means that we can activate LPMO in the photo by photoactivation, and that can be used in a number of applications. Well, we're structural molecular biologists, so we like looking at the structures, we decided to have a look at the low resolution structure of these two-domain LPMO. No two-domain LPMOs have been studied structurally before. Again, the same problem. We have a long linker between catalytic domain and cellulose binding modules. That makes a crystallization of the full domain enzyme very difficult, if possible at all. So we decided to have a look using SACS, and we retrieved the low resolution model of the enzyme, and it indeed has a huge lobe, a big lobe, in which catalytic domain in the actual model of the catalytic domain can be accommodated, and smaller lobe in which cellulose binding module can be docked, and a linker by a peptide will just pass from the catalytic domain to the cellulose binding module. Well, LPMOs serve to catalytically nick cellulose chain and activate cellulose reactions. So we ask, okay, which kind of cellulases they will most efficiently synergize with? And the answer is GH7, actually, the one which I showed at the beginning. If you test GH5, 7, 12, and 45, which are the most common families of cellulases, fungal cellulases, you see that there is only one strong synergetic partner, GH7. We don't know yet why it is so. That's something to study. Then we said, okay, well, it works with the cellulosic substrates. Can we have a look at the real biomass? And we looked at the LPMO activation of the commercial enzymatic cocktail at liquid hot water pretreated sugarcane bagasse, steam exploded sugarcane bagasse, and dilute acid pretreated sugarcane bagasse. All of them were the hydrolysis of all these three substrates were activated, but in different degree. Steam exploded was the best substrate for cellulose activation. And that brings us to another point, which I think is important. From my point of view, we always need to study physical structure of the insoluble substrates, because reaction between enzymes and insoluble substrates happens at the interface, and what is this interface will strongly influence the outcome of the enzymatic activity. So we established a set of physical techniques to study, to look at the plant biomass, and this scanning electron microscopy images of the sugarcane gas untreated, and after acid pretreatment, you see that the gas changes. And when you apply alkaline pretreatment as a second pretreatment step, you can see the cellulose bundles which will be liberated, and that's when enzymatic hydrolysis is the highest. We also looked at the biomass using solid-state NMR, and you not only can follow the decrease in concentration of lignin and correlate the cellulose accessibility with efficiency of enzymatic hydrolysis, but also calculate crystallinity. And actually, if you do so, you can show that solid-state NMR crystallinities are quite a bit more reliable than X-ray diffraction. Crystallinity is determined from the X-ray diffraction experiments. If you add necessary corrections to the X-ray diffraction crystallinities, you can come to the parity close to the solid-state NMR ones. We also looked at the water relaxation times inside the bagasse and their dependence on the pretreatment, and actually T2 values, relaxation times, they report on water interactions with the cell wall, basically. And one can see that when this inverse of the relaxation times comes to the plateau, it's when efficiency of enzymatic glycolysis becomes very, very high. So basically, it is a measurement which can be used to follow or to predict the possible efficiency of enzymatic glycolysis. We also looked at the biomass using confocal images, and one can see that not only color of the biomass changes with pretreatment, but also average decay time of the lignin autofluorescence also changes. And there is a huge distribution in decay times correlated with the nature and condensation of lignin. That has direct effects on the enzymatic glycolysis and the pretreatment. So I believe that physical studies of the insoluble substrates in the area of one biomass, for example, is very important for developing efficient pretreatments and pretreatment and enzymatic glycolysis technology. Okay, well, that's where we are now. Now, what else? What next? Cellulose ethanol is being produced at the industrial scale. What we can do in addition to that with plant biomass? Well, we can do quite a lot, actually. We can use it as material, for example. If you pretreat grasses, you can see these cellulose fibers which appears, needles. And that's because cellulose comes in fibrils and which assemble themselves in fibers. And one can disassemble its fibers. When we produce sugars from cellulose, we actually chop all these fibers into glucose. If you don't do that, you can end up with nanocellulose or microfibrillated cellulose, which is very, very strong material. And one can do it enzymatically. Here we compare enzymatically produced microfibrillated cellulose with temp oxidation produced cellulose. You can see that cellulose fibers are way longer. That's a distribution of the measured fibers. Not only that, they are also quite a bit more thermoresistant as compared to temp oxidation. I believe that enzymes will be more and more used in material research and production of materials. Well, we also can produce prebiotics. You probably know that we have billions of bacteria in our guts, and some of them are good, some of them are bad, and some of them are ugly. And the good bacteria helps us to have a stronger immunological response, lower our probability to have cancer, and in generally improve our style of life. Bad bacteria cause dysentery or in war situations, incurable bacterial infections like Clostridium difficile is really tough. One can produce prebiotics, the oligosaccharides, which will activate probiotic bacteria from plant biomass, for example, from xylem. And xylem comes in two types, arabinoxylem and glucuronoxylem. And if we take xylanase, for example, and apply them to xylem, we will produce oligosaccharides, and those will have prebiotic potential. If we take, well, we produced an structure of this particular xylanase from J10 family and show that it is not very efficient in cleaving xylotriose, but it's very efficient in cleaving xylohexose, pentose, and tetrose. And that is good because it is known that short oligosaccharides, they have a high prebiotic potential. And then we applied it to biomass, and one can see that from biomass, it will liberate a range of oligosaccharides, which actually with time will converge to shorter oligosaccharides like cellulose. And it does it with a relatively good efficiency. It also stimulates enzymatic cocktail. Curiously, it not only improves xylem hydrolysis, but also improves cellulose hydrolysis, probably because it will clean the surface from cellulose from the xylose bound to it. We saw the structure of the enzyme to try to rationalize why does what we studied in our biochemical work of the research, and that's the shape of this enzyme. That's the product of the reaction bound to the active center. We looked at the active site, and it has three product subsites and three substrate subsites, in total 6 subsites. That's an active site addition. And we looked at the decorations and in which of the subsites and explain why it does what it does. So without further ado, and before I will be switched off by Eduardo from access to the internet, I will go to the conclusions. Our LPMO is a two-domain LPMO with C1, C4 rigid selectivity. Contaminations from LPMO fungal production can be eliminated by thermal treatment, denaturation, and renaturation of LPMO. And actually, that's a universal approach. It can be done with practically any resistant LPMO, which again is quite good. The enzyme can be activated by light in the presence of chlorophyllin and ascorbic acid, and we definitely would like to study in more depth light activation of LPMOs. This LPMO has considerable synergy with endoglucanases, but specifically with GH7 endoglucanases and not with endoglucanases, fungal endoglucanase. It also boosts enzymatic, commercial enzymatic cocktail, and that effect will depend on the type of biomass pretreatment. The low resolution shape of the enzyme has a oil-like shape with a big domain, big catalytic lobe, and smaller cellulose binding module, and joint binding, very long linker peptide. There is a growing gap between our knowledge of sequences of the existing enzymes and their activities and functions. Actually, I think that's a huge, huge scientific and biotechnological opportunity which we could explore, because for the first time in our lives, we can choose any amount of possibly potentially interesting enzymes to fiddle around and to study and eventually to apply in biotechnological research. I'm certain that more new and novel enzymes will be discovered and characterized in the future. Physical structure of the substrates, from my point of view, is absolutely necessary in our studies of enzymatic degradation of insoluble substrates. Simply because that's a heterogeneous catalysis, enzymatic catalysis, and what happening in the surface is of outstanding importance for these kind of catalysis. And finally, abundance and low prices of biopolymers, cellulose, hemicellulose, starch, chitin, and agricultural residues, and also renewable nature of these substrates call for establishing biotechnology of their transformations and fuels of food ingredients and renewable materials, which is basically building bioeconomy, and that's what I believe we should Well, with that, I would like to thank all of you for your attention and organizing for giving me this opportunity to present some of our results. Thank you very much.
Thank you, Igor, for the nice presentation and for sticking to the time. Thank you. We have now time for questions. And actually, we have one question here that was posted in the question and answer by Monisha Christian. She asks, does substrate utilization by microbes in separate cases of bacteria or fungi affect the types, quality, purity, and or perhaps the toxicity of metabolites that it produces by bioconversion in either bioremediation or biodegradation aspects?
Right. Really general, important general question. Well, the short answer is yes, of course. But what I would like to point out is that I think it's crucial to understand how the enzymes interact with substrate, because that's what you can improve rationally if you understand what the enzymes do individually and characterize them and then apply them to hydrolyzed biomass. I mean, obviously, if we are talking about using of microorganisms, it becomes a little bit more complex, because the whole internal metabolism of the cells will come into play. So I guess it's easier to start by studying individual enzymes, composing them in possible enzymatic mixtures, and then applying them to transform biomass, and then slowly build up to more complex systems, basically going from biochemistry to synthetic biochemistry to synthetic biology, then rather trying to solve problems of the synthetic biology or microbiology of microorganisms. So that's my opinion.
Great. Is there any other question? People can also either write their questions or raise their hands. Oh, there is one. Hobson, please.
Hi, Igor. Thank you very much for your presentation. I also work with recombinant protein production, but I'm wondering, it is very difficult to produce GH7 and LPMO proteins. And how do you guys manage to produce those proteins using E. coli as a host? Do you have any tip for us?
Yeah. I mean, if we are talking about fungal proteins, then my tip, try not to produce them in E. coli. We actually started our activities in structural investigations of GH7s, for example, trying to express them in E. coli, and it doesn't work, because fungal proteins frequently are strongly have a number of disulfide bonds, which are not very well formed by E. coli, and they're also glycosylated, which will not happen in E. coli. So if you want to produce fungal cellulases, you have to produce them either in fungi. We use Aspergillus nigrans, but also Aspergillus niger or Trichoderma rhizi or whatever, or yeast. P.K. pastoris is another possibility. They will differ in glycosylation and efficiency of production, but that's the way to go. That's why we had to establish expression systems in different microorganisms, because bacterial cellulases can be efficiently produced in E. coli. Now, fungal cellulases, you will have difficulties, I'm afraid, when trying to express them in E. coli. So the same applies to LPMOs. If you want to express LPMOs in E. coli, look at the bacterial homologs, and chances are high. All the LPMOs which we studied and also fungal cell lyses were expressed in fungi in our lab.
Okay, thank you.
Are there any other questions? So if not, so we're going to move on to the next talk. It's going to be presented by Dr. Abe